good morning, everyone. So it's our great pleasure to have Professor Amy Rosenzweig from Northwestern University to conduct this distinguished seminar in the Faculty of Science, which we started last year, initiated by Professor Guo and the Faculty of Science. So first, may I invite Professor Guo to introduce Amy, please.
Okay, thank you, Hongja. Yeah. Well, good morning, afternoon, or evening, whoever or wherever you are. It's my pleasure to welcome the Faculty of Science Distinguished Lecture today, entitled Copper-Dependent Biologic Methane Oxidation. Under the mounting threat of climate change, increasing atmospheric methane concentrations are a constant source of concern and debate. The second most abundant greenhouse gas, of course, after carbon dioxide, methane accounts for at least 25% of the current global warming or climate change causing molecules. Conversion of methane to desirable fuels and chemicals could simultaneously mitigate global warming at the same time meet increasing energy demands, or chemical demands for that matter. However, current catalysts that can selectively activate this almost like 105 kilocal per mole CH bond, we know the bond energy, in methane molecules require high temperatures and pressures, along with the significant capital expenses. In nature, aerobic methane oxidation is accomplished under ambient conditions by bacteria called methanotrophes. In most methanotrophes, this chemically challenging reaction is catalyzed by a particular methane monooxygenase, which we call the PMMO, which is a copper-dependent integral methane enzyme, or membrane enzyme. So PMMO is composed of 3 subunits, which Professor Rosenzweig will explain further, which are arranged in a trimeric complex. So despite expensive research or extensive research and the availability of multiple crystal structures, the location and nature of these copper active sites remain controversial. Studies are further complicated by issues with retaining enzymatic activity and uncertainties regarding the possible involvement of additional protein components in a complex, as we know, enzymatic structure. So the quest to address these issues will be discussed. So it's my great pleasure today to introduce our speaker, Professor Amy Rosenzweig today. Amy is a Weiberg Family Distinguished Professor of Life Sciences at Northwestern University. She received a BA in chemistry from Amherst College and a PhD in inorganic chemistry from Massachusetts Institute of Technology. After postdoctoral research at Harvard Medical School, she joined the faculty of Northwestern University. She's a fellow of American Academy of Arts and Sciences and a member of the National Academy of Sciences. Her laboratory uses structural, biochemical, and omic approaches to attack problems at the forefront of bio-inorganic chemistry. Areas of interest include biologic methane oxidation, metal uptake and transport, and oxygen activation by metal enzymes. So without further ado, I would like to welcome to the floor or to the Zoom Professor Rosenzweig. Please.
Thank you.
Thank you.
All right, let me share my screen. Okay, can everyone see okay? Good? Okay, great. Well, thank you for inviting me to give this lecture. And it is 9 o'clock at night here. So if I say anything silly, it's just because I'm not used to giving a talk at 9 p.m. on a Sunday night. So my lab works on bacteria that convert methane to methanol called methanotrophs. And we have several different projects in the lab focusing on these bacteria. I'll just talk about one today. So shown here is the metabolic pathway of these bacteria. So these bacteria live on methane gas as their sole source of carbon and energy. And in the first step of their metabolism, which we'll be focusing on, they convert methane to methanol. And then the methanol is further converted onto formaldehyde and formate and carbon dioxide. So these bacteria are abundant and they're found in many different environments. One of the strains we work on in our lab, Methylococcus capsulatus bath, was first isolated from these hot baths in Bath, England. There's a lot of different strains that are found in soil and rice paddies. There are some found in water, all different types of environments. And even recently, methanotrophs have been discovered living in extreme environments, such as these volcanic mud pots in Italy. And these types of methanotrophs live at very low pH and very high temperatures. But at Northwestern in my lab, they can be found growing in our fermenter here. And so we're blowing methane and oxygen gas into this fermenter, being very careful not to hit the spontaneously combustible range. And then we're isolating the enzyme that we work on directly from these methanotrophic bacteria. So why should anyone be interested in bacteria that eat methane? There are several reasons. It turns out that there's a lot of methane gas that's just wasted. So this is a satellite view of the Midwest of the US. This is where I am right now in Chicago, so where all these bright lights are. And up here in North Dakota is the Bakken Shale, where there's a lot of hydraulic fracturing or fracking going on. And at sites like this, tons of gas is just being burned and wasted to the tune of millions of dollars per month. And that's because methods to turn methane gas into methanol and then on into more easily transportable fuels are expensive and require high temperature, high pressure, and big catalytic facilities that are hard to build. And it's not just that we're wasting methane, but methane, as was mentioned in the introduction, is a potent greenhouse gas. And so that's not such a good thing. This is, I just took this from the New York Times. This was an accident that happened in Ohio where so much methane gas was released into the atmosphere, they judged it to be equivalent to something like a whole year's worth of methane emissions in all of Europe. And so people have taken some crazy approaches to trying to mitigate methane emissions. So this is an example of a US patent where they propose to construct a vessel on the back of a cow, and then cows breathe out a lot of methane. So the idea is that you would put the very bacteria we work on, the methanotrophs, inside here, and they would capture all the methane before it goes on to cause global warming. But having grown these bacteria in the lab for years and years, I know that they're very difficult to grow, and it would be totally ridiculous to try to grow them on cows. Other ideas have more to do with not necessarily catching the methane, but making animals not emit so much methane. So that's a big problem in Australia. They have lots of livestock emitting methane. And so these are some sheep where they are have apparatuses on them to, they are feeding them some kind of vitamins so they don't give off so much methane, and they're trying to see what they do. So there's lots of people from diverse fields interested in methane, and one thing to do with all this methane gas would be to turn it into something more useful. And there are lots of synthetic biologists who have devised plans and pathways to engineer methanotrophic bacteria. The idea being that you take all that carbon from methane, it goes to methanol, and then you engineer the bacteria to do all sorts of fancy pathways in sticking in genes to make different compounds that would be useful. So things like butanediol are very popular. But the problem with all of these pathways is that while you can install these pathways in methanotrophs, or you might think about putting the methane oxidizing enzymes in other hosts, it all hinges on understanding this first step, the oxidation of methane to methanol. So there's no point in engineering your methanotroph to take methanol to other things if you can't turn the methane into methanol fast enough. And the methane monooxygenases actually aren't that fast of enzymes. So it's really important that we understand this first step, and that's where our research is. So we're trying to understand in methanotrophs this very first step: how do they convert methane into methanol? And it turns out that methanotrophs have two different solutions to carrying out this reaction. So here's the reaction in a little more detail. It's a monooxygenase, so it takes one atom of oxygen to make methanol and the other goes to water, and it requires 2 electrons and 2 protons. And the bacteria have two different solutions to this. So there's 2 different methane monooxygenase enzymes. There's a soluble methane monooxygenase shown here, which is actually an iron enzyme. So it uses a dinuclear iron center shown here to convert methane to methanol. And this enzyme is only produced by a subset of methanotrophs, and it's produced when they're starved for copper. So when they have no copper around, they use this iron enzyme. The majority of methanotrophs, however, produce this membrane-bound enzyme called particulate, or PMMO. Particulate just means membrane-bound. And there are lots of methanotrophs that only make the PMMO. And then in the ones that can make the SMMO, as soon as they get enough copper, which is very little copper really, in their growth environment, they make the PMMO. And out there in nature, there's usually enough copper to make the PMMO. So this is the prevalent one in nature. When the methanotrophs are making the PMMO, they also manufacture these extensive intracytoplasmic membranes. So this is a picture of one cell. And so all of these membranes are filled up with the PMMO. You can see that better on this slide here. And so that's actually really interesting. The regulation of how these membranes are formed depends on copper. And even though we've known about this membrane formation for 40 or 50 years, we still don't understand exactly how copper causes it to happen. But for us doing biochemistry, it's very helpful, because what it means is that the bacteria kind of naturally overproduce the PMMO because all these membranes are filled with it. So if we grow the bacteria in the fermenter, isolate the cells, break them open and isolate the membranes, we can get a lot of enzyme to work with. And that's a good thing, because nobody's ever been able to express PMMO or SMMO for that matter in a heterologous host. So we can't express this in E. coli or anything else. So it's good that we can get it from the native organism. The drawback is that there's no easy way to make site-directed mutants. And you'll see that's been kind of a problem for understanding the biochemistry. So we solved the structure of PMMO for the first time about 15 years ago, and I'll talk a lot more about the structure, but this is the overall architecture. So it's a trimer, and it's made up of 3 copies each of 3 different subunits. It has an extensive periplasmic region. So these are gram-negative bacteria. So this part sits in the periplasm. And then it has a very extensive transmembrane region that's housed in these intracytoplasmic membranes, or ICMs. So I'll break down the structure in for you on this slide. So what I call the protomer is 1/3 of that trimer. So all PMMOs we've looked at have the trimer. This is just 1/3 of it. And the protomer is made up of 3 subunits. PMOB is shown here in gray, and this consists of 2 paraplasmic cupridoxin-like domains, and then 2 transmembrane helices. PMOA, shown here in this pink color, is all transmembrane helices, as is PMOC, shown here in purple. And we've determined the crystal structures of PMMOs from a range of different methanotrophs. So here are some of the other structures that we've obtained. I'll point out that this PMOC subunit, shown here in purple, seems to be the most flexible. So its conformation changes a bit in the different structures. And in the case of this structure here, it's actually primarily disordered. We can't even see it in the electron density. In this structure here, from this organism called species Rockwell, sorry, my pointer is getting stuck. There's also this helix shown in yellow that we call helix X, and that's not attached to the PMOC subunit. So it appears to come from some other protein that may co-purify with the PMMO. And we've tried very hard to identify this through different mass spec approaches. And we still don't know what that helix is, but since we're purifying the enzyme from the native organism, it could be something important. So we're interested in figuring that out. So that's the overall architecture. Now, PMMO has a long history of debate about the nature of its metal active site. I think everyone always agreed it was a metalloenzyme, but it's long been debated whether it uses copper, whether it uses iron, how many of each metal ion it uses, and so on. And I certainly won't rehash the history. I'll just tell you that I think everyone now agrees that it's a copper-dependent enzyme. And shown on this slide is an example of an experiment that we did, and that we did many times to address that question. And so what we do is we take what I call the membrane-bound PMMO, and that's the isolated membranes from the organism before we solubilize with detergent or purify or anything. And those membranes are probably 80% PMMO. And we use those for studies that involve measuring enzyme activity, because those are more active than the purified protein. And what we do in these types of experiments is we treat the sample with cyanide to remove all the metals, and that abolishes the activity. So on this plot here, we have no metal present and we have no activity. And then when we add copper back, we can recover activity, and that maxes out at about 2 coppers per protomer, which is the structural unit I showed you on the last slide. If we add other metals such as iron, we can't restore activity. So the activity is dependent on copper. Now, when we keep adding copper, the activity actually becomes completely inhibited. We're still not sure exactly how that happens, but what happens is that after you abolish all the activity with copper, you can actually take that material, retreat it with cyanide, and then add copper again and get activity back. So it's reversible. I'll also point out here that these 2 experiments are done with 2 different reductants: duraquinol, which is a mimic of endogenous quinols such as ubiquinol, and NADH, which is also able to provide reducing equivalents to PMMO membranes. The physiological pathway for reducing PMMO is still under debate. But we do need to reduce the enzyme, presumably because we need to make a copper-1 species that reacts with oxygen to make whatever species reacts with methane. So it's a copper enzyme, and knowing that, we can look in the crystal structures and see what types of metals we find and try to determine where the active site is in this structure. And so when we solved the structure at first, so this first structure from M. capsulatus bath, we found several different metal sites, and I'll go through those. The first site, which we call the bis-his copper site, is located here in between these two paraplasmic domains. And in that site, we have a single copper ion coordinated by two histidine residues, His-48 and His-72. His-48 is not a strictly conserved residue. So in some PMMOs, it's actually an asparagine. And if we look at the structures of those PMMOs, this is an example of one here, we see no metal bound in that site. So that suggests it's not a likely candidate for the active site. Moreover, even in some PMMOs where the two ligands are conserved, we don't see any copper in that site. And so that's detected by, in the crystal structure by anomalous data collected at the copper absorption edge. So that site is either not conserved or not always occupied. So again, unlikely to be the site where methane and oxygen bind. The second copper site I'll tell you about is what we call the copper B site. And that's located here in the PMOB subunit, right at the interface with the transmembrane region. And in our original structure of PMMO, we modeled that with two copper ions, as shown here. There are three histidine residues surrounding this site. The first one, His-33, is the amino terminus of the PMOB subunit. So right there, the first 32 residues are a signal sequence that gets cleaved off. And then there are these two other histidine residues found in an HXH motif. And in the model, the amino terminal nitrogen is also within coordinating distance. Now, the reason that we modeled this as dicopper originally is because we had collected a large amount of extended X-ray absorption fine structure data. An example is shown here, which suggested that in all of our samples of PMMO, there was copper interacting with another copper at a short copper-copper distance of 2.5 to 2.6 angstroms. That's this feature here in the Fourier transform of these data. However, as we went on and got more crystal structures, it became clear that this site in the structure was really best fit with a single copper ion. So here's that same site in another structure that was higher quality and slightly better resolution. And it's the same three ligands, but best modeled for coordinate with a single copper ion. And here's another example where we see the same thing. And this is actually our highest resolution crystal structure thus far. All right, so I'll come back to the discussion of that site in a minute, but let me tell you about the third metal binding site that we found in the crystal structures. And that one is located within the membrane in this PMOC subunit shown in purple. In the first structure of PMMO that we got, that site was actually occupied by zinc. And it turned out that that zinc came from the crystallization solution. So we can only crystallize that particular PMMO with zinc. And that's because zinc was also found elsewhere mediating contacts in the crystal lattice. However, PMMO as isolated does not contain any zinc. There's no relation between zinc and PMMO activity. So when we crystallize without zinc, which we were able to do for some of the PMMOs from other organisms, we saw different things. In this structure, we actually saw no metal in that site, and we have a detergent molecule kind of wedged up in there. And you can see that those three ligands are sort of moved apart because that detergent molecule is in there. I also had pointed out on an earlier slide that the PMOC subunit changes conformation, and in this structure, that's because there was that detergent in there pushing these helices apart. Now, in the one structure where we didn't have an issue with zinc or detergent, this structure here from Methylcystis species strain Rockwell, we actually found copper in that site. So to our best guess, we believe that copper is what's supposed to be in there, physiologically speaking. However, there's a big issue with this copper C site, and that's shown here. The copper C site in the structure is right next to a region of the polypeptide chain that we can never see. So in this structure, there's about 2830 residues between 198 and 226 that are disordered, meaning there's just no electron density for these residues, so we can't model them. And that's the case in all of the crystal structures of PMMO. This is the sequence that we're missing in this structure. And note that there are a number of potential metal binding residues here. There's a couple of glutamates, or 3 glutamates. There's a couple of histidines. And so without seeing what that loop or whatever it is looks like, I kind of feel like we don't exactly know what's going on at that site. And that's become more and more disturbing to us over the years, because what's happened is as more and more genome sequences have become available, we've looked at more and more sequences and done a lot of bioinformatics. And that region of PMOC that we can't see turns out to be the most highly conserved part of the subunit. So this is a sequence logo for the entire PMOC, and the 1-letter codes for the residues that are most highly conserved are the bigger ones. And this region stands out as having a whole huge long stretch of highly conserved residues blown up here. So it seems that the most highly conserved and perhaps most important part of this subunit of PMMO is we haven't been able to visualize yet. And I'll come back to that. So one big problem with this enzyme is that I already told you it's not a super fast enzyme to start with. This is the turnover number of cells producing PMMO. And when you start taking PMMO out of the cells and then taking it out of the membranes and purifying it, you lose a lot of activity. And that's not unusual for membrane proteins. A lot of times when you take a membrane protein out of its native environment, its function can be harmed. But that's a problem if you're not totally sure that the enzyme that was used to grow the crystals and gather all these structural data represents the active form of PMMO. So we're looking at all these metal centers trying to determine which is the active site. We really would like to know if those metal centers that I just described to you are also present in the active PMMO in the cells that are actively turning over methane. And so we, a few years ago, decided to take a step back and try to really address that question, being, are the same copper sites present in vivo? And one way we did this was through EPR spectroscopy that we did in collaboration with Brian Hoffman's lab at Northwestern. And we had a joint student, Matt Ross, who did this work. And what Matt did is he grew the methanotrophs in the fermenter on N15 and copper 63. And shown up top here is an EPR spectrum of the whole cells. And so that spectrum, so EPR gives you a readout on any species that has unpaired electrons. So by EPR, we can look at copper 2, but not copper 1. And the cells show a nice EPR signal that's attributable to a single copper 2 species that I'm calling copper B, and you'll see why in a minute. When he took the cells, broke them open, solubilized the PMMO with detergent and purified it, he got this blue spectrum here. And you can see that the spectrum becomes more complicated, and that's because now it's a mixture of 2 different copper 2 species that I'll call copper B and copper C. So the copper C site or signal is not present in the whole cells, presumably because it's copper 1. And then once you break open the cells and start manipulating the enzyme, some of it oxidizes, and that's why you get this mixture. So because you only see the single signal in the whole cells, we were able to get some more information about the coordination of the copper 2 giving rise to that signal. And here Matt exploited the fact that he was using, that his sample was labeled with N15, and he was able to analyze the hyperfine couplings on these lines. This is a second derivative. Anyway, just take my word for it that it fits with this equation to tell you that there are 4 nitrogen ligands on the copper that's giving rise to this copper 2 signal that we see here. So whole cells have one type of copper 2 coordinated by 4 nitrogens. We were also able to then use ENDOR spectroscopy, which is kind of like a combination of EPR and ENDOR, to get more information on these nitrogens. And what ENDOR is really good at is looking for histidine coordination, because in ENDOR you can get signals from both nitrogens that are directly coordinated to your copper 2 species and nitrogens that are distal, meaning if you have a histidine ring, you can figure out how many nitrogens are coordinated, and you can also see the signals from the nitrogens on the other sides of the rings. So the black here are those whole cells that we call the vivo PMMO. And the ENDOR spectra from that through Gaussian fitting and quantitation gave 2 different types of nitrogens with an area ratio of 1 to 3, basically suggesting or telling you that you have 4 nitrogens, which is what the hyperfine analysis on the last slide showed you. And then this part is interesting. The part of the experiment that detects the distal nitrogens was fit with an area ratio of 1 to 2, basically saying there are 3 distal nitrogens there. So if you have 4 nitrogens and 3 distal nitrogens, that means that 3 of your nitrogen ligands come from histidine and the other comes from some other nitrogen, whether it's this amino terminus or backbone nitrogen. So this is very useful because there's only a single site in PMMO where 3 imidazoles from histidine can simultaneously coordinate copper 2, and that's this copper B site, which is why I was calling that EPR spectrum copper B. So this tells us that this site that we see in all the crystal structures is also present in vivo. So that is very good to know. It also confirms that that site is mononuclear because of the 4 nitrogen coordination. Now, if I go back to that other signal, oops, sorry, this copper C signal, we weren't able to do the same kind of detailed analysis because we can't isolate that signal from the copper B signal. So while we can see that that signal, which would be evident in these ENDOR spectra here, we can see that it adds something on top of the one from the copper B. We can't isolate it itself, but it does tell us that there is also nitrogen coordinated to that site. So here's our model for that. And Matt also has some data I won't show suggesting solvent there. So we weren't sure where that site is coming from, although from the naming you can gather that I'm going to say it's this site here. But since we were using samples that also had this bis-his site in them, we wanted to figure that out. And the way we did that was using another fancy paramagnetic resonance technique called DEER that can give you the distances between 2 EPR active species. And we worked with Fraser Macmillan at the University of East Anglia in the UK for this. And we took advantage of the fact that PMMO is this big trimer. So there's all these different copper-copper distances you could be looking for because this technique looks for long distances. And based on those data, we think that that copper C signal, that second signal, is coming from this site here. So all of that taken together tells us that the sites that we're looking at in the structure are most likely present in vivo, and we're not, you know, way off the mark trying to think about the active site by looking at the structures. And It's not like there's some huge multinuclear cluster in PMMO that we've purified away that we're missing out on. So then that brings us back to this question of why is the activity of PMMO so poor if we seem to have the same copper centers in vivo, more or less, that we have in our purified enzyme that we used for the structure. And we thought that some of the problem might have to do with taking PMMO out of its native membrane. To address that question, we took PMMO and we reconstituted it into micelles. So the samples that we had mostly been working with were in detergent micelles, and that's also what we crystallized, this protein detergent type of aggregate. In a micelle, you surround the membrane part of the protein with lipids, and then you have detergent around that. So it's more like a membrane-bound environment. And so we, and I'll just point out also that when we were doing these experiments, we also changed our methane oxidation assay so that we only use C13 labeled methane and then detect the labeled methanol that's produced. And that's important because methanol is a common contaminant in lots of buffers and all sorts of things. So if you look at our activity numbers down here, if you just measure methanol production by GC, you get higher numbers than if you use you measure the labeled methanol production by GCMS, and the samples are the same. It's just that you're getting rid of that background. And that's important since our activities are pretty low to start with. If you're looking for small amounts, you want to be sure that it's actually coming from your enzyme and not stray methanol in your solutions. So here are some experiments where we measured the methane oxidation activity in the membrane, in the solubilized sample, and then in the micelles. And there's a lot of data here, so let me just draw your attention to this here. Here are some solubilized samples in detergent, and when we take that and put it back into the micelle, we recover a significant amount of activity. Again, the assays are being performed with two different reductants. Similarly, if you have the purified protein, you can recover activity in the micelle. This plot here just shows that the copper content is not changing. So we can recover a reasonable amount of activity with the same amount of copper in the sample. So that's telling us that the loss of activity is not due to loss of copper, or maybe a little bit due to loss of copper, but there's a big contribution from the membrane environment. These are similar experiments for another PMMO that has very bad activity. So if you look at the solubilized and purified samples, they're completely dead. And at least using the duraquinol reductant, we can recover activity, again, with similar copper concentrations. So the membrane is very important for the activity of PMMO. So having established that it's reasonable to look at the sites in the crystal structure to try to address the nature of the active site, let's talk a little bit more about these sites. This copper B site, again, has the 3 histidine ligands. And while those ligands are conserved in most PMMOs, so the top part here of these sequence alignments, and I'll point out that these sequences also include AMO-B subunits. AMO is ammonia monooxygenase, and it's a homolog of PMMO. It's even very poorly studied because it's even more difficult to handle than PMMO. So while these histidines are conserved in all of these, the so-called type 3 methanotrophs, which are the methanotrophs that were found in those volcanic mud vents that grow at low pH and high temperature, those sequences actually completely lack the 3 histidines. So they have a different residue at the amino terminus, either isoleucine or methionine, and they have a PVG sequence. So if those PMMOs have the same type of active site as the rest of the PMMOs and the AMOs. Perhaps this is not the active site. There's only about 8 sequences of these thus far, and there's no biochemistry on these bugs. They're very difficult to grow. So we don't have much information yet. That's something we're pursuing now. In contrast, the ligands to the copper C site are highly conserved. So the 2 histidines and the aspartic acid, and those are highlighted in purple here, and those are conserved in all of the sequences, as is that disordered region that I showed you earlier. So unfortunately for us, that PMOC site, while being the most conserved, is the one that we know the least about, that we're least certain of what's going on. And that's again, because in one structure we had zinc, in another we had detergent in there, in another we had copper. That's what I just showed. And then in this one, as I mentioned earlier, it was so disordered that we couldn't model anything in that site. So we wanted to turn to other techniques to try to get more information about what's going on at this copper C site. And we decided to start with this PMMO from this organism we call 20Z, since we had the least information. And we decided to use a technique called native top-down mass spectrometry to look at the 20Z. And for this experiment, we had to reconstitute the protein in nanodiscs. So this is another way to mimic the membrane. We again surround the protein by lipids, but instead of putting detergent around the lipids are surrounded by a membrane scaffold protein, or MSP. So you make this disc to put the protein in. And kind of crazy, but to do this native top-down mass spectrometry technique, you need to put your membrane protein in nanodiscs, even though what you're going to do is shoot it into a mass spec and break everything apart. And so what this technique does is it takes the whole complex. You can actually detect the mass of the whole complex, and then you can detect masses of protomers, and then you can detect the different subunits. And then if you're lucky, you can fragment the subunits. And this is very powerful because you can actually localize metal ions to different subunits. And if you're lucky, you can localize them to even pieces of sequence in the subunits. So instead of doing something like ICP-MS, where you just say, okay, I have 3 coppers in my protein complex, you can actually say, I have 3 coppers and they're located in these particular subunits. And so for our problems with the PMOC subunit, we thought this would be really great because we could see if it had copper, see how many coppers it had, and maybe fragment it and find out something about that disordered region. It didn't work out quite as we dreamed, but I'll show you what we did find out. So here are our masses of the different subunits. So the MS2 shown here. We got a good mass for PMOA. For PMOB, we got a good mass with a single copper. So very consistent with what we see from the structures and the spectroscopy. And then for PMOC, we got kind of a mixture of species. We got intact PMOC mass with no metal. And then we got this truncated PMOC with just a few residues missing off the amino terminus, completely unclear why, and just a small amount of that species had copper in it. So this was very messy, and we were particularly worried about the truncation and this very small amount of copper. However, we went on and tried to get the fragmentation, the MS3, and these are just the data for PMOB. No surprises here. This fragment underlined in green that has the HXH sequence has the copper in it. So just more verification. But we weren't able to get this to work for PMOC. And it turned out that we were losing copper when we reconstituted the nanodiscs. And so here's what we had when we started in detergent for this PMMO. And when we put it in the nanodiscs, we lost copper. So we wondered if we were losing copper specifically from the PMOC subunit. And to investigate this further, we did some experiments using a different PMMO. So the 20Z PMMO has very low activity. So we switched to the species Rockwell-pMMO. And so here's a more detailed analysis of what happens with copper when you put it into nanodiscs. So here's the membranes that have about 3 coppers per protomer. When you put it into the nanodiscs, you lose copper. But we found that if we added copper during the preparation of the nanodiscs, we could get more copper in this sample. And the reason we switched to this PMMO is because it has better activity, and we wanted to correlate this with activity as well. So here are the corresponding activity numbers, the membranes, the nanodiscs, and then the nanodiscs when we include more copper when we make them. And this was very striking to us because we get this big improvement in activity when we do the nanodisc reconstitution with copper. We then looked at these samples by native top-down MS. So this is the sample in the nanodisc when we don't add the copper in the reconstitution. And again, we see this business where PMOC is truncated and we have very little sample with copper. However, when we add copper during the nanodisc reconstitution, the truncation is gone. I have no idea why, but most importantly, we get much more full-length PMOC with copper in it. And that we thought was really exciting because not only were we seeing the copper in that subunit, but we had shown that that correlated with an increase in activity. And that's really the first indication that that site in the PMOC subunit is important for activity. It's kind of suggesting that maybe that site could be the active site. So what are we doing now? So since we put PMMO into the nanodiscs for the mass spec, we are now pursuing cryo-EM of PMMO and nanodiscs. And these are just some initial images. So you can kind of nicely see the PMMO in the nanodisc. And I don't have the structures to show you yet, but the goal here is to visualize the portion of PMOC that we can't see in any of the crystal structures, get a better look at that copper C site, and maybe understand what the membrane is doing to help the enzyme to be active and perhaps to stabilize that copper C site. So that's where we're going with this. So just to summarize, PMMO has 2 monocopper sites in vivo and in the crystal structures. This suggests that methane oxidation occurs at a monocopper site. Increased copper occupancy of the PMOC subunit, presumably in that copper C site, can be correlated with restoring enzymatic activity in the nanodiscs. And that suggests that the copper C site is important for activity and may very well be the active site. So we have a lot more to do. I'll just acknowledge the lab in our Zoom picture here. We have our personalized lab masks that say, "I love Methanotrophics." So some of the work I described was from former lab members such as Raquel Lieberman and Sarah Sarajadan, Steve Smith, Rama Balasubramanian. The nanodisc work and the ongoing cryo-EM work has been done by Chris Koo. Sue Rowe, who left recently and is not in the picture, did all of the BICEL work that I described. Frank Tucci is working on preparing native lipids, and Jerry Roberts is working on some new PMMOs from other organisms. Ricky Jotz is a joint student with Brian Hoffman, who is following up on the EPR work that Matt Ross started. And the rest of these guys are working on other projects related to copper homeostasis and methanobactins. And I let's see, I talked about Brian Hoffman. The native top-down mass spec was of course done with Neil Kelleher and his student Louise Schachner, also at Northwestern. Yuan He is a cryo-EM assistant professor who's been advising us on cryo-EM. And thank you very much for listening, and I'd be happy to answer any questions.
Okay, thank you, Amy. Very exciting talk. And now, so all of the participants, and you can send your questions and through the chat. And so colleagues in the panels, then you are, of course, more than welcome to ask questions and directly.
Maybe thanks again, Professor Rosenzweig. My question is, clearly it's very important to understand the structures of the enzyme. Now, the query I had was how or maybe your group or other groups looked into the actual diffusion channels for methane, because that may help maybe understanding of the mechanisms or active sites, and even the probably functionality of membrane. Just a thought, yeah.
Right, so methane should be more soluble in the membrane. So you might think that methane enters the enzyme from some aspect of the membrane, and then perhaps the methanol that's produced might go out into the periplasm, because that's where the methanol dehydrogenase enzyme is. So we've certainly tried to analyze the structure for channels and cavities. Not really finding anything. It's sort of, that doesn't seem to work so well on the transmembrane parts, because there's lots of hydrophobic areas, and then there's that piece of the Pmoc subunit that's disordered. So I wouldn't say, we haven't found anything obvious in the structure that's a path for the methane for how it gets in there. And we don't have any data, unfortunately, where we have any hydrocarbons bound, or a lot of times people put like xenon in crystal structures to try to look for oxygen or methane pathways. We've never been successful at doing anything like that. So tough to say.
Yeah, thank you for that. Yeah, good elaboration.
For the copper C site, are the ligands in a facial coordination geometry? I'm not sure what you mean by facial, like on one side, or? Who asked that question?
It's from the audience, is it?
Can I ask a question?
Oh, they can't answer. Oh, they can't speak.
Can I ask a question?
Yeah.
Okay. Thank you for the talk. And I'm curious about the substrate specificity of this enzyme, because so if you can use some other substrate, then it may be also like useful for investigating the mechanism. So I'm curious about this point.
Yeah, so the PMMO is active with substrates up to at least 4 or 5 carbons long, possibly longer, but the literature from years ago suggests not much longer. It's not as versatile as the soluble MMO. So the soluble MMO can hydroxylate aromatics and larger substrates. The PMMO can't. That may provide some insight into the previous question about how the substrate gets in. We've not been able to crystallize with anything like that, or get any evidence from spectroscopy of things binding. A lot of the work on the SMMO used radical clock substrates and different types of substrates to try to investigate the mechanism. Depending on which of those substrates we could get our hands on, we could maybe do that, but you have to have a pretty active enzyme to do things like that. So, but yeah, it's definitely something we're always thinking about.
Okay.
Thank you.
So, Klembo, you may
see.
ask questions directly.
Okay, yeah, hi, Amy, excellent talk. I'm wondering, probably you mentioned that you do not know the physiological cofactor for PMMO. Any guess? Do you see any like NADH binding site in your protein and things like that?
Yeah, so I don't think there's an NADH binding site. So the thinking with NADH is that in the samples where you can use NADH as a reductase, there's probably some sort of an NADH oxidoreductase a little bit in the sample. And then that is reducing quinones to quinols, and they're perhaps directly reducing the PMMO. So that's generally what people think about using NADH, and then that would suggest that the quinol might directly interact with PMMO. There are several models in vivo though, so that's one of them. There's another model that suggests that the electrons actually come back from methanol dehydrogenase through a cytochrome, through its acceptor, a cytochrome C. And there's a few papers where people have done metabolic modeling where they predict what's going to happen with these different pathways, and they got different answers for the different methanotrophs. So, but
Sorry. Yeah, that's very interesting. So does it mean that the secret, unstructured region, which is also very conserved region, could it interact with those unknown, electron donor that have yet to identify?
It could. So one interesting thing about it is the enzyme is in this trimer, and that disordered region is in the middle of the trimer. So like, would the reductant be in there in the middle like that? I just don't know. That seems weird to me, right? Because if it's diffusing laterally through the membrane at quinol, it seems weird that it would somehow get stuck inside there, but who knows?
Is there any, is the quaternary structure of the PMMO has to be always trimeric? Do you see any chance of dissociating? No chance.
It's always a trimer.
Okay. Obligation, okay. Fine. Thank you.
Yes.
Thanks, excellent talk anyway. Yeah.
Sure. Let's see.
Yingwu, do you want to ask questions directly?
Yeah, thanks. Hi, very nice talk. So I have two questions. So first is, because the CAP-C site is the active site, then my question is, what's the role of the CAP-B site? May it electron transfer some role?
Yeah, great question. Well, first of all, let me just say, we don't know for sure that the copper C site is the active site.
Okay.
The copper B site, I don't know, it doesn't really look like an electron transfer site, and there's no evidence that it undergoes redox chemistry. However, we haven't proven that it's not an electron transfer site, and it's weird that it's not conserved in that one set of sequences. I think it does. I think it has a function, though. I'm just not sure what it is. Another option is that it's structural for some reason. I guess the short answer is we don't know. We've often thought of electron transfer, but we have no proof of that.
Okay, nice. So my second question is for the CAP-C site. Do you have any observed the oxygen binding or the methane bound form, a substrate binding form in the crystal structure?
No, I mean, well, it would be very hard to observe methane since it's just a single carbon, but no, we haven't. And that site, because it's so near a disordered region, it's generally not looking very good. But no, we've never seen anything anywhere near that site.
Okay. From your publication, I found that if you say that the oxygen binding may generate the hydroxide radicals, so that will be the active species for the reaction. So if you could detect the hydrogen radicals using, for example, EPR techniques to find evidence, that may be quite good to provide direct evidence for the capsaicide, right?
Yeah, that would be great if we could detect any sort of intermediates or anything.
Yes.
Yeah.
So thank you a lot.
Yeah, thanks.
Okay, we have one more question from the audience. And
Here's a question. Should I answer the one in the chat?
Do we know anything about the evolution of these bacteria and their potential role in shaping the Earth's geochemical cycling?
Well, that's a big question. I mean, there are a number of papers about, you know, their sequences and stuff like that, but their potential role in shaping the Earth's geochemical cycling. I mean, all I can say is they're aerobic, so they came after the great oxidation event, and they, you know, they're methanogens, which are these anaerobic bacteria that produce methane, and so then the methanotrophs are located just above them and eat a lot of that methane. So they came after, I guess, but yeah, I guess I'm not really sure how to address that.
Can I ask a question?
Sure.
Hi, Amy. I have two questions. First of all, so in the mass spec of the nanodisk complex, I noticed that when the complex depleted with copper, you see truncated PMOC, whereas if you supply the copper, you see the full length. Does it mean that copper actually plays a structural role to, you know, for the integrity of the protein, stability and integrity of the protein?
Yeah, maybe. It's weird because it's only, I think it's only like 5 residues off the terminus, but yeah, that's a good point.
Okay.
Only
about 5
No,
amino
I
acids.
actually haven't thought about that in a while, so that's a good point.
Okay.
Thank you.
Another question is about the genetics of methanotrophes. So why these organisms are difficult in genetics? They're difficult for DNA uptake, or low or poor homologous recombination, or?
Yeah, so it is possible to, depending on the strain, to make knockouts. We've done that. It's possible to make mutations. It's all very slow because they grow very slowly, so it takes a really long time. We have been able to mutate a few residues, but the problem is you can't mutate any residue like that's involved in metal binding. We've not been able to get those mutants, and you'd think that you should be able to do that by using a strain that you can grow on the iron containing MMO, right?
Yeah.
It doesn't work. Nobody's been able to do it. It's almost like they still need the PMMO, even if they're not going to use it. So it's and there, you know, and there are a number of geneticists and microbiologists, Mary Lidstrom, there's the group in England, Colin Morrell, so people who actually do this for a living and have been struggling for years. And sort of, I guess another approach that's been taken is, you know, expressing them in other methanotrophs. And yeah, it's all very, very slow, and particular difficulties with the PMMO. And there is actually one paper about a CRISPR-Cas9 system in the methanotroph, but the efficiency was so low that it doesn't seem like it's going to be very tractable.
Okay, thank you.
And I have a question. Hello, can I have a question? So I'm wondering about the copper B and copper C site. So from the mass spec experiment, it seems that the copper can be easily come out from the copper C site. So my first question is, does it mean that the copper B site bind copper stronger than the copper C site?
Yeah, I don't know because of the, I mean, yeah, that's definitely what it implies. Unless it has something to do with the copper C site being within the membrane and close to the lipids. And so somehow it's more easily disruptible. But it most certainly appears that way. From everything we've done, the copper B site is always nice and well occupied, and the copper C site is much more labile. But you think if there's an active site there, though, it should bind reasonably tightly. So I'm just not sure if it also has something to do with the membrane.
Yes, that's my second question. So you mentioned that you can remove all the copper by treating the enzyme with cyanide, and then you put in different equivalents of copper in to restore the activity. And then you find that copper, the activity is maximum with 2 copper. So I'm wondering if you could put another metal specifically in one of the sites and to check the activity.
Yes, maybe we could. We haven't really played around with that, but yeah, that's definitely something we've thought about and talked about and just haven't done. I mean, we've sort of done something like that when we did a bunch of experiments with zinc and tried to figure out where the zinc went. We published a paper on that 6 or 7 years ago. But it wasn't quite as systematic as what you're suggesting. That's a good idea.
See, thank you.
Okay, I may ask several questions. And I'm not sure, have you done any mechanistic study on the role of individual residues in the copper binding site? And
we can't get mutants. We cannot mutate any of the residues in the copper binding sites. If you try to mutate those in the methanotroph, you can't. It doesn't work. You can't. The bugs don't live.
oh, I see. And I remember that Sunny Chen synthesized some model compounds to elaborate the role of this MMO. So I'm not sure any comments on his work and in terms of the protein structure and function. And
so, you know, I don't know about those small molecule synthetic compounds. I can't really comment on those, or whether, I'm not sure they oxidized methane. Maybe they did. I think we agree with Sunny that it's a copper enzyme. We have not seen evidence for the specific copper site that he proposed, the tricopper site in the PMO-A subunit. We have not seen evidence through crystallography for a metal binding site in PMO-A. So I'm not sure if he still believes that the copper site is there. And we have not seen evidence for a tricopper site, which is what he proposed. I think we agree that it's a copper enzyme.
okay.
And yeah, I can't address the models or he did some peptide work as well.
Yeah, and so one of your EPR, you use the whole cell to demonstrate the copper binding. It's really very impressive. I assume normally the membrane protein, the expression is, the yield is low.
This protein is very highly expressed, so
okay.
we're very lucky. The membranes are just chock full of the PMMO. It's estimated to be like 20% of the proteome of these bacteria. So we're very lucky in that respect.
Oh, okay. So would that be possible, for example, if you overexpress the protein with limited copper, or maybe copper rich, to see any differences in terms of copper, for example, binding to the protein, or whether one side is occupied by the copper and the other side probably not? I don't know the technical details, probably you can.
Yeah, Yeah, that's definitely possible. One thing we've been interested in for a long time is investigating, especially when they're starved for copper and they're still making some PMMO, what if there's anything in it. That's definitely possible.
Okay. So any other questions from the panel? Okay, Chen.
Oh, thanks for the fascinating talk. So my question is, does zinc inhibit the enzyme? Because one of the structures you have zinc sort of competing with copper.
Yes, we can definitely see inhibition by zinc.
Oh,
Okay.
So, and we know that zinc goes into that site. But we also did some experiments a while back where we could see zinc. We soaked in zinc and we could see some zinc going into the other site as well. But yeah, zinc does inhibit.
Yeah, it seems to have a sort of higher affinity than the copper because it replaces.
Yeah.
Thank you.
okay, any other questions from the panel or audience? So I try to see. Okay, I think if no, we shall stop this seminar.